All right. Hello, everyone. My name is Nicole Sherloc, and I'm the Invitae lead for APAC in Canada. So on behalf of everyone here at Invitae, thank you so much for your time and welcome to our webinar titled Invitae Exome Interpretation, Leading Science and Innovation to Bring Exomes to Scale. Before we get started, I'll quickly run through a few housekeeping items. Firstly, all participants are muted. However, I strongly encourage you to submit questions at any point along the way during the presentation using the questions section on your webinar control panel. And we will do our best to address as many questions as possible at the end during the Q&A. Today's webinar will also be recorded and will be available on the Invitae website soon. So our first speaker will be Dr. Heather McLaughlin. Dr. McLaughlin is a board-certified clinical molecular geneticist with clinical expertise in cardiogenetics, connective tissue disorders, neurogenetics, and pediatrics. Before joining Invitae, Dr. McLaughlin was an associate director of clinical genomics and cardiogenetics at GeneDx, where she was instrumental in developing their clinical exome sequencing program. Dr. McLaughlin obtained her PhD in human genetics from the University of Michigan and completed her clinical molecular genetics fellowship at Harvard Medical School. Our second speaker will be Amy Knight-Johnson. Ms. Knight-Johnson is a board-certified genetic counselor, and before joining Invitae, she was a senior genetic counselor at the University of Chicago Molecular Diagnostic Laboratory and has experience providing clinical genetic counseling services in prenatal and pediatric settings. So Dr. McLaughlin will start off our presentations today by discussing the challenges of exome sequencing and how Invitae's clinical exome analysis combines both automated gene and variant curation together with expert evaluation. Ms. Knight-Johnson will then follow as a second presenter, and she will discuss a few case examples to really demonstrate Invitae's approach to exome analysis. And with that, we'll begin the presentation with Dr. Heather McLaughlin. Please note it will take a few seconds for us just to switch presenters, so please bear with us.
Thank you, Nicole, for the introduction. I'm really excited to share with everyone today the cutting-edge enhancements we've made to our exome platform that enable us to provide affordable, comprehensive exome sequencing to all patients, and how these enhancements allow us to launch a first-of-its-kind case-level exome reanalysis program that is included in the cost of the test. So what is exome sequencing? As opposed to panels that target a set of genes related to a condition or phenotype in question, exome sequencing simultaneously interrogates the vast majority of all coding exons. This ends up being about 250,000 exons, or 1.5% of the genome. By identifying changes in the DNA across the exome, we can offer a comprehensive analysis to pinpoint an individual's genetic diagnosis. There are several situations when exome sequencing might be considered for a patient. The first is when their condition is associated with many genes, and this can be the case for situations involving intellectual disability and neurodevelopmental conditions. The second example is when a patient is a product of a consanguineous union. And in this situation, exome sequencing allows us to simultaneously interrogate many genes associated with autosomal recessive disorders. Exome sequencing may also be considered for situations where a patient has multiple diagnoses or if there's involvement of multiple unrelated organ systems. And again, in this situation, the list of differentials can be rather large. Finally, exome sequencing is often recommended for patients who have already undergone a diagnostic odyssey without a definitive diagnosis. So perhaps previous genetic testing has been done before or been negative or inconclusive, or other diagnostic assays has been unable to identify a diagnosis. Many studies have now been published in the literature demonstrating the impressive yield of exome sequencing, as high as 20 to 40%, in patients with neurodevelopmental disorders, multiple congenital anomalies, and syndromic clinical presentations. Although exome sequencing has been instrumental in the diagnosis of individuals with rare genetic disorders, there are several challenges that stand in the way of its full implementation in clinical care. The first is the constant evolution of gene phenotype information. Almost every day, novel disease genes are published in the literature, and the phenotypes that are associated with known disorders is also constantly expanding. Because of these issues, it's quite difficult to perform a reproducible and unbiased evaluation of genetic variation in the context of the proband's phenotype. The interpretation of exome data requires manual review and judgment of variance in gene-disease relationships, and bias does exist due to the level of experience and knowledge that the individual performing the analysis has on various genes and disorders. We also know that there's a need for consistent evidence-based gene disease curation and variant interpretation frameworks. In order to perform a complete analysis, we need to have up-to-date gene, disease, and phenotype curation information, and also a semi-quantitative variant interpretation framework that is consistent with the American College of Medical Genetics and the Association for Molecular Pathology guidelines. While the guidelines that exist today are an excellent framework, it is our experience that refinement is required for clinically meaningful variant interpretations. Finally, one of the most difficult issues to address in the age of clinical exome sequencing has been developing a reanalysis framework that allows laboratories to routinely apply learnings from newly published information and internal data to help solve previously negative or inconclusive exome cases, and more importantly, to do it at scale. So in order to address the challenges of exome sequencing for rare genetic conditions, Invitae's clinical exome analysis combines automated gene and variant curation with expert evaluation for fast, reproducible, and accurate exome interpretation. This slide outlines the different components of Invitae's exome sequencing test, and in the subsequent slides, I'm going to share more detailed information about our state-of-the-art interpretation process. Shown at the top of the diagram in teal is the wealth of information that gets fed into Invitae's Moon. Moon, which is shown in blue, is Invitae's intelligent engine that integrates and weighs genomic and phenotypic data. At the bottom of the figure in green is an outline of our reporting process where our expert clinical staff ensure accurate, clinically relevant reporting for each and every patient. Clinical information is important for a phenotype-driven test such as exome sequencing. So in the next couple of slides, I'd like to review with you Invitae's clinical data gathering and portal capabilities that help clinicians to ensure that a complete clinical picture is obtained for all patients undergoing exome sequencing at Invitae. Here is a screenshot of our ordering portal. Our exome can be ordered as a Proband-only test, as a duo test with a proband and an affected parent, or as a trio. I do want to say here that trio testing has been shown at our lab and other laboratories to produce the highest yield. You'll see at the bottom of the slide here that each sequenced individual is given the opportunity to opt in or out of secondary findings analysis, which focuses on 59 genes associated with clinically actionable genetic disorders. We determine which variants to report based on the molecular strength of the variant and the clinical fit of the gene-disease associations with the patient's phenotype. Therefore, it's important that we receive as much clinical information about the patient as possible. To that end, we provide multiple ways for providers to submit clinical information. Shown here is another screenshot of the portal. At the very top of the screenshot, you can see where free text can be entered for additional clinical information. And we also provide various mechanisms to upload previous results and family history. So for example, you might be able to copy and paste a recent clinic note into the box, and we have natural language processing tools that systematically identify human phenotype ontology terms from your clinic note. We are also able to obtain pedigrees and family histories, upload previous genetic testing results, and also any previous diagnostic results that the patient has undergone, for example, an MRI, EEG, etc. We also have a number of online ordering resources for the clinician. The first shown here is our gene coverage tool. This allows clinicians to review any technical limitations for the genes of interest before ordering. For example, you might be able to review if a gene has low coverage, or if there are any CNV gaps that are present before the gene, and this can all happen before ordering. Within the portal at the time of ordering, clinicians are also able to specify any genes of interest that might be on their differential. And in addition, clinicians can also provide us with additional phenotypic information using our HPO selection tool. So here, clinicians can enter their own HPO terms. You can search this tool using features by organ or system. And I do want to note here that the clinic notes that are provided in the ordering portal at the time of ordering are also reviewed for each case by our experts to add more HPO terms prior to the exome analysis. Now, the quality of the data produced for clinical exome tests is really the key to an accurate result. And so in the next few slides, I'm going to review Invitae's exome assay performance with respect to detection of single nucleotide variants, indels, and copy number variation. First, I'm going to briefly discuss the basic steps to a next-generation sequencing assay. First, the genomic DNA is extracted from the patient sample. That DNA is then fragmented into smaller pieces, and we can use capture probes or baits to bind and catch the exonic DNA. That captured DNA is then sequenced, and the outcome is a bunch of small reads or copies of that DNA. These reads are then bioinformatically aligned to a reference sequence, and the variants are called. These detected variants can then be interpreted, classified, and finally, when appropriate, reported to the provider. At Invitae, our exome assay offers comprehensive coverage of over 20,000 genes. We also have enhanced coverage of over 7,000 disease-associated genes, and 99.4% of the reportable range, which is generally considered to be exons plus or minus 10 base pairs, is covered at 20x. The average coverage is 150x. In terms of the types of variants that we can detect, of course, single nucleotide variants, small deletions and insertions, less than 50 base pairs, and copy number variants may also be detected with high sensitivity at greater than or equal to 4 exons. Although I do want to say smaller CNVs, even single exon CNVs, may also be detected and reported. Importantly, the turnaround time for our test is 6 to 8 weeks, and 95% of our cases are currently being reported on time. Invitae's exome test also allows for sensitive detection of intragenic copy number variation by next-generation sequencing. So the uniformity of coverage in next-generation sequencing tests is very important when it comes to designing high-quality assays. We know that the read depth can vary among exonic regions, but it can be reproducible amongst samples. We can see this in the example below, where each line represents a sequencing depth profile for an individual sample. We see that there's variation between coverage from exon to exon, but we can also clearly see a reproducible pattern of this coverage between samples. This uniformity that we see here is key to allowing for high-resolution intragenic CNV calling, and that's made possible by Invitae's state-of-the-art lab automation efforts. By testing many different samples, you can start to see patterns and create a baseline of depth that you normally would observe in any region, and then we can look for deviations with respect to the baseline samples. If in any particular example, for example, you see more reads or depth of coverage than you normally would observe, then that could mean that there is a duplication in that patient sample. And conversely, if you see less reads or coverage in that region, that may indicate that a patient has a deletion in that part of the genome. By using this technology, exome sequencing can simultaneously identify both sequence variants and intragenic CNVs with high sensitivity from a single sample. In the next few slides, I'm going to describe how Invitae's MOON utilizes data from Sherloc, our validated and quantitative variant classification platform. So as I discussed in previous slides, there are many challenges in germline genetic variant interpretation. Like many other labs, Invitae adheres to the framework put forth by the ACMG and the AMP. However, there is persistent concern of discordance between and within clinical laboratories resulting from things like inappropriate application of variant interpretation rules, inconsistent use of evidence among scientists and laboratories, and finally, insufficient granularity for categorizing the evidence. Sherloc was created to try to address some of the issues that exist in interpreting germline genetic variants. Sherloc utilizes a linear process of variant interpretation that involves highly trained scientists, genetic counselors, and laboratory directors. It is a validated and quantitative variant interpretation method that ensures transparency and consistency, and it also occurs in an auditable, custom-built software platform. Sherloc was created on the bedrock of the 28 rules that were established by the ACMG and AMP guidelines. In the course of over 7 years and 15 revisions, after interpreting variants in over 11,000 genes and interpreting over 790,000 interpreted variants, we have created over 200 detailed refinements to the ACMG and AMP rules. Sherloc considers 5 types of evidence in a hierarchical approach. The first is population data, the second is variant type, the third is clinical observations, and these are both clinical observations that we see internally during testing at Invitae and also externally in the literature. The fourth are experimental studies, and the fifth are indirect and computational evidence. We utilize a rule-based scoring system for each individual piece of evidence using either benign or pathogenic points. And these points are used to, are applied to a point-based score threshold to determine the final classification based on the ACMG suggested 5-tier classification system. So next I wanted to provide you with a little bit more detail about the types of evidence that we use in variant interpretation. So for population data, you can see that benign or pathogenic points are awarded depending on the frequency or the rarity of the variant in population databases. We also take a look at sequence observation, so the different types of variants that we see, whether the variant is missense or perhaps a predicted loss of function variant. We also take a look at various types of clinical findings. So was the variant determined to be de novo? Do we see the variant in individuals that have very specific phenotypes for a gene and disorder in question? If the variant is occurs in a recessive gene, do we see maybe 2 variants that are in trans? And then we also take a look at segregation data. In terms of molecular studies, we do a thorough and rigorous analysis of the studies, and depending on the type of study that is performed and the evidence, we can apply either strong or weak evidence towards the variant either being neutral or causing a disrupted protein product. Finally, we take a look at computationally predictive evidence, trying to determine if the evidence points towards the variant being neutral or deleterious. In terms of evaluating molecular studies and computational predictions, one difference at Invitae is that we use a validated functional modeling tool to better inform these criteria for missense variants specifically. Invitae's functional modeling platform, or FMP tools, use training data to create gene-specific models that can accurately inform whether specific missense variants are pathogenic. For in silico tools, we have sequence and biophysical analyses. For indirect evidence, we can use spatial analyses. And for experimental evidence, we have molecular and cellular assays. All of these together allow us to test hypotheses about the impact of missense variants on both protein structure and function. I do want to note here that unlike existing computational approaches that have a single model for all genes, Invitae's functional modeling platform is trained to evaluate variants in each gene separately, and that allows us to take into account disease-specific domains and molecular mechanisms, allowing for a much more accurate classification. One of the things that makes Invitae's functional modeling platform so powerful is that we have instituted prospective performance of our FMP tools using unknown variants. So I'm going to show 2 examples here. In example A, the variant was entered into ClinVar as a variant of uncertain significance in 2018. Later that same year, our FMP tool predicted this particular variant to be damaging. And in 2019, another independent laboratory classified this variant as pathogenic in ClinVar. So this essentially supports that our predicted damaging evidence generated by our FMP tool was accurately predicting the variant to be deleterious. The example here in variant B, in 2018, the variant was not present in ClinVar, but our FMP tool predicted that the variant would be benign. And then in 2019, an independent laboratory classified the variant as benign in ClinVar, again, giving us confidence that this tool is accurately predicting the variant to be benign. Importantly, in a set of over 700 variants for which our FMP tools predicted either damaging or benign evidence, and an independent classification of the variant was later uploaded into ClinVar, we found that our tools accurately predicted damaging or benign evidence over 95% of the time. And because of this, we use prospective performance to test all of our FMP tools, and this is an important part of the assessment for when a tool meets our accuracy standard and can be used in clinical variant interpretation. Invitae's variant interpretation process considers information from both public databases, like ClinVar, for example, and also Invitae's internal database. Importantly, at Invitae, over 1 million tests have been performed in many different disease areas, and we use this wealth of knowledge to provide more accurate variant interpretations for all of our patients. We also believe that at Invitae that data sharing is critical among laboratories. For those that aren't familiar, ClinVar is a freely accessible public archive of reports of the relationships between human variation and phenotypes with supporting evidence. And as of August of 2020, Invitae was the top submitter to ClinVar with over 392,000 submissions. So now I'd like to switch gears a little bit and talk more about Invitae's intelligent engine Moon and how it works in combination with phenotypic information to return a list of suspicious variants for expert review. Recently, Invitae acquired Diploid, the maker of Moon and Apollo software. In the next few slides, I'm going to discuss some exciting enhancements to our exome and assay as we integrate these softwares into our exome analysis platform. The Moon platform has a proven history of providing rapid and accurate results. Moon has been reported in multiple manuscripts, including studies from Rady Children's Hospital, where they utilized Moon to interpret whole genome sequencing data from patients in the NICU. Moon was shown to find 99% of the causative pathogenic variants identified via expert manual review in 95 children. This software was also instrumental in Rady's Guinness World Record for genome analysis. Moon is an intelligent engine that integrates and weighs genetic and phenotypic data to identify suspicious variants. Apollo offers continuous weighted gene phenotype curation from scientific literature. And in a pilot study of 150 previously solved Invitae Exome cases, Moon alone correctly identified 97% of the causative variants in less than 2 minutes per case. So much like Rady's experience, Invitae also found high precision compared to expert manual analysis. Next, I would like to describe how the Moon platform works. Input into Moon is raw data from the patient in the form of variant call files, any HPO terms describing the patient's phenotype, the patient's biological sex and age of onset of features, and also information about the family, including whether there are other affected individuals in the family. What Moon does is weigh variants based on public data sources like Nomad, ClinVar, and dbSNP, internal data sources, for example, variants that are previously interpreted at Invitae or Sherloc variant interpretation framework criteria. And it also considers information from Apollo, which again is a gene-disease relationship network curated daily from a combination of automated literature searches and manual expert quality control of gene-disease relationships. So the result here is an intelligent engine that integrates and weighs molecular effect, scientific evidence, and clinical phenotype to identify suspicious variants for a case. Apollo, again, is a continuous weighted gene phenotype curation that occurs from the scientific literature. So the literature is continuously searched, and through natural language processing, HPO terms are extracted from the literature. There is then a manual curation and quality control of the findings. So for every gene disease association, phenotypes are listed, but also ranked based on the frequency in which that feature is associated with the gene disease in the literature. We are also working to include our own internal cases to further refine the databases. This happens real-time with the emergence of new literature and therefore provides a real-time, meaningful gene disease curated database. Currently, the Apollo database has quite high-quality phenotypic information for more than 6,000 gene disease relationships. This ever-growing knowledge base integrates new scientific insights in real time, which guarantees a comprehensive and up-to-date analysis for every patient. One of the most important parts of Invitae's exome sequencing interpretation is our clinical team and the expert review that they provide for each and every case. In the next few slides, I'm going to talk about our expert team and also the results that you can expect from obtaining exome sequencing at Invitae. Invitae's clinical team of exome experts is comprised of an experienced team of PhD scientists, certified genetic counselors, and American Board of Medical Genetics and Genomics certified laboratory directors. We have broad experience in clinical and academic laboratories. And importantly, we have sequenced several thousand probands and over 10,000 individuals who have been sequenced via our exome test. Our team of exome experts aims to provide a personalized report with clinically relevant variants. To help us, Moon provides an objective list of suspicious variants for manual review, and Sherloc enables accurate classification of the reported variants. Our reports include pathogenic variants, likely pathogenic variants, and variants of uncertain significance in genes when phenotypes and inheritance patterns match the known gene disease associations. We also report single pathogenic and likely pathogenic variants in autosomal recessive genes when there is a strong clinical overlap with the patient's phenotype, treatment is available, or when further testing might rule the condition in or out. Overall, our goal is to identify a variant in a gene or genes with strong clinical overlap to the patient's indication for testing. So now we've reached perhaps the most exciting part of the talk today, where we're going to provide an overview of Invitae's reevaluation and reanalysis programs. There have been several recent studies that have shown the importance of case-level exome reanalysis, and the ACMG recently issued a statement outlining points to consider in the reevaluation and reanalysis of genomic test results. Following the adoption of clinical next-generation and exome sequencing, the rate of discovery of novel disease gene associations skyrocketed in the early 2000s. And while the rate of novel disease gene discovery has slowed in recent years, it still remains over 500 genes per year. And what I would like everyone to consider here is that anytime a patient is sequenced and then years go by where many different new novel disease genes are discovered, that sort of represents a missed opportunity for that patient to achieve a molecular diagnosis. So it's very important that exome data be reconsidered over time. Before I move on to talk about Invitae's reevaluation and reanalysis programs, I would like to provide clarification of these two terms. So the first term, variant level reevaluation, occurs when a report is updated report is issued because of known variant upgrades or downgrades, or when a gene is now considered a disease gene, whereas before it was considered a candidate gene. These types of reevaluation cases are identified through internal databases based on changes to a specific variant or gene, and then the corresponding cases are reviewed and reports are updated. So this is a pretty narrow focus, the variant level reevaluation. On the other hand, case level reanalysis is a re-review of all variants that were identified in a case, both reported and unreported in the context of the patient's phenotype. And this can also include maybe new or updated phenotypic information that wasn't available at the time of the first analysis. Many labs do state that they do a continuous reanalysis in the background, but really what they're talking about here is variant level reevaluation, not really case level reanalysis like we're talking about here. Case level reanalysis has demonstrated to lead to an increased yield of up to 20%, with the highest yields achieved over 1 year after the first analysis. So at Invitae, we are committed to achieving high diagnostic yields for all of our patients. And so we have instituted both reevaluation and reanalysis policies that I'd like to share with you today. The first, variant level reevaluation, is something that Invitae has always done and will continue to do, where variants are reevaluated when new data become available. The second, case level reanalysis, is a new policy that I'm introducing to everybody today. And this is an expert case level reanalysis that can include any new clinical information that you provide every 6 months for a minimum of 3 years. When we do a case level reanalysis, any new positive findings are reported, and clinician requested reanalysis may also be available. This expert case level reanalysis that occurs on a 6-month cadence puts less burden on the patient and provider. So there is no need to remember to request a reanalysis or decide on the best time to ask for one. This ensures that patients receive the most up-to-date information about their genetic data. Case level reanalysis is offered at no additional charge, as it is included as part of the original order of the test. So next I wanted to address how Invitae is addressing some of the challenges that we talked about in the beginning of the talk. So first, you might remember we talked about this constant evolution of gene phenotype information. We are tackling this issue by using Apollo, which again is a continuous weighted gene phenotype curation that is occurring from scientific literature. Natural language processing is used to scan the literature, and importantly, it's followed by manual expert quality control of gene-disease relationships. We also talked about difficulty in performing a reproducible and unbiased evaluation of genetic variation in the context of a proband's phenotype. And today we presented Invitae's MOON, where we integrate and weigh information like molecular effect, scientific evidence, and the patient's clinical phenotype to identify suspicious variants for each case. We also talked about the need for a consistent evidence-based gene disease curation and variant interpretation framework. And I explained today about Invitae's use of Sherloc, which is our automated, consistent, and comprehensive refinement of the ACMGIAMP variant classification criteria. And finally, we talked about performing reanalysis at scale, and I just presented to you Invitae's policy of offering expert case-level reanalysis for all patients every 6 months. Thank you again for allowing me to present the enhancements we've made to our exome assay that enable us to provide affordable and comprehensive exome sequencing and a first-of-its-kind case-level exome reanalysis program. Now I'm going to turn it over to Amy Knight-Johnson, who's going to discuss a few case examples that demonstrate Invitae's approach to exome analysis.
Hello, and thank you for the introduction. I'm excited to share some case examples with you today to help illustrate how the MOON software integrates into Invitae's exome platform in order to provide a personalized and comprehensive exome analysis. The first case I'm going to present today is a 1-year-old male who was referred for Proband-only exome analysis and was noted to have multiple findings, including microcephaly, developmental delay, encephalopathy, hypertonia, failure to thrive, and dysmorphic facial features. For this case, MOON generated a ranked list of 6 suspicious variants in 5 genes. So this is the short list of variants considered most likely to be causative for the patient's phenotype based on MOON's integration of the genetic and phenotypic data for the case. The variant that was ranked top by MOON in this case is a hemizygous missense variant in the MECP2 gene. In the center of the slide, you can see the patient features listed, and as part of the ranking algorithm, MOON looks for phenotypic overlap between the patient and the phenotypes that are curated for that gene-disease association in the Apollo database. The patient clinical features that are highlighted here with a check mark are those where there is overlap with phenotypes curated for MECP2 in the Apollo database. So in this example, we see that the patient's features have significant overlap with what's been described in other individuals with MECP2 related conditions, with 8 of the 10 reported features for this patient having overlap with MECP2 based on the curation in Apollo. So now let's review MECP2 in more detail. MECP2 is located on the X chromosome, and many of you may be familiar with this gene as associated with classic Rett syndrome in females, and in males, this gene has an association with a severe encephalopathy that has onset in the neonatal period. And pathogenic variants in this gene are typically de novo. So, because of the differing presentations in males and females for this gene, the associated phenotypes are curated as separate conditions in the Apollo database. On the right of this slide, you can see some of the most common features described in males with MECP2 variants, and these are listed in order based on the frequency with which each particular phenotype has been described in affected individuals. So for this condition, for example, encephalopathy and hypotonia are the most commonly reported symptoms. In this slide, we're just showing a snapshot of the most commonly reported features, but in the Apollo database itself, there are additional features curated that are just less commonly observed. The Moon algorithm not only considers which of the patient's phenotypes overlap with the curated condition in the Apollo database, but when ranking variants, it also considers whether the patient is noted to have features that are commonly observed in that condition. So here we've highlighted with a star the most common features associated with MECP2 variants in males that were also reported in this patient. And you can see that the patient is noted to have multiple features that overlap with the most common features observed in other affected individuals. However, you'll also note that there are some common features, such as myoclonus and seizures, that are not noted for this patient. So we do know that many features of different conditions may develop over time, or that some affected individuals may never develop a specific feature of a condition, even if it's a common feature of that disorder. Therefore, while we're assessing the phenotypic overlap, Moon doesn't require a perfect match for any given condition in order to rank a variant as suspicious. Once Moon has ranked the variant and the phenotypic overlap has been assessed, the variant is then interpreted using our Sherloc interpretation framework. In this case, our interpretation included a manual review of the published literature, including one case that had the same variant as was seen in our patient, and also additional reports of different variants impacting the same amino acid. We also look at data on population frequency and in silico predictions. We also assess additional information such as clinical data and inheritance from our internal observations. In this case, which was a Proband-only exome, after the initial analysis was completed, the mother was later tested and the variant was found to be de novo. So all this evidence contributed to a pathogenic interpretation for this variant. In this case, the original Moon shortlist included 6 variants, including the MECP2 variant. So based on our interpretation of that variant and the degree of phenotypic overlap, the MECP2 variant is considered to be the cause of the patient's phenotype in this case. However, we know that some patients have a dual diagnosis, so a further review of the other variants is still needed. So here I'll present a couple of examples of the other variants that were ranked for this case and show how they were assessed. The Moon shortlist for this case included 2 heterozygous variants of uncertain significance in the CPT2 gene, and this gene is associated with autosomal recessive carnitine palmitoyltransferase 2 deficiency, or CPT2 deficiency. The image on the right highlights with a check mark the features that are noted to be present in this patient that are also reported in CPT2 deficiency. And so you can see that the patient does have some clinical overlap with the condition, including microcephaly, hypotonia, and feeding difficulties. CPT2 deficiency is a condition that has variable severity, including a neonatal onset form of the condition. On the right of the slide, you can see common features observed in individuals with CPT2 deficiency from the Apollo database, and the ones highlighted with a star are those that are also present in this patient. So notably in this case, although there is clinical overlap between the patient and CPT2 deficiency, the patient lacks many of the other common features of the condition. The two variants observed in this case are present at a low frequency in the population and have not previously been reported in affected individuals and were interpreted as variants of uncertain significance. So overall in this case, based on the degree of phenotypic overlap and the presence of a pathogenic variant in MECP2, our expert clinical team determined that these variants were not likely to be contributing to the patient's phenotype. Another variant that was ranked by Moon for this patient was a heterozygous missense variant in the NFIA gene. This gene is associated with an autosomal dominant condition that's characterized by macrocephaly and brain malformations. The image on the right highlights with a check mark the features that are noted to be present in this patient that have also been reported in individuals with NFIA-related conditions. So this patient does have some phenotypic overlap with this gene, including hypertonia, developmental delay, failure to thrive, and dysmorphic facial features. The variant identified in NFIA in this case was not present in population databases and has not previously been reported in affected individuals. On the right of the slide, you can see some common features observed in individuals with NFIA-related conditions from the Apollo database, and highlighted with a star, you can see the features that are also present in this patient. And so, although there is some overlap, the patient is not noted to have some of the most common features of the condition, including brain malformations. And in addition, this patient was noted to have microcephaly, whereas macrocephaly is a common feature of NFIA-related conditions. Overall in this case, based on the degree of phenotypic overlap and the presence of a diagnostic finding in MECP2, our expert clinical team determined that this variant was not likely to be contributing to the patient's phenotype. However, it's important to also bear in mind that as a part of our manual review process, we do take into consideration whether we can fully evaluate for all the features associated with the condition. In this case, for example, that included taking into consideration the lack of brain MRI results for this patient that would have been necessary in order to identify the associated brain malformations. Our second case example is a 3-year-old male who was referred for Proband-only exome analysis and was noted to have a history of developmental delay, hypertonia, failure to thrive, ataxia, nystagmus, and cerebellar hyperplasia. In this case, Moon returned a ranked shortlist of 11 suspicious variants for our expert team to review. The variant that was ranked first in this case was a homozygous missense variant in PMM2. Here we see the clinical features noted for this patient, including a check mark to highlight those which have overlap with features reported in individuals with PMM2-related conditions. In this case, the phenotypic overlap appears to be strong, with the features that are noted for this patient having almost complete overlap with PMM2 based on the Apollo database curation. PMM2 is associated with autosomal recessive congenital disorder of glycosylation type 1A, or CDG1A, and affected individuals with this condition have a type 1 transferrin isoform pattern when you do carbohydrate deficient transferrin analysis. On the right, you can see the most frequently reported features in individuals with CDG1A, and those highlighted with a star are those that are also noted to be present in this patient. So in this case, not only have the patient's features been associated with CDG1A, but they're also common features of the condition. Once a variant has been ranked by Moon, it's then interpreted based on our Sherloc interpretation framework. This includes a manual review of all published literature for that variant, in addition to considering factors such as variant effect, population data, and in silico predictors. This variant is described as a missense, but we know it occurs in the last nucleotide of the exon, and so is predicted to impact splicing. And it also has a low population frequency of 0.002%. Although there was limited published information about this variant, Invitae had multiple internal observations of this variant in individuals that had a type 1 transferrin isoform pattern. So based on these internal observations, we were able to reach an interpretation of pathogenic for this variant, and as this variant was observed in the homozygous state in this patient, the finding was consistent with a diagnosis of CDG1A. In this case, the patient was 3 years old when they were referred for exome analysis. They had already developed multiple clinical features associated with CDG1A, and they also had brain MRI results available that help contribute to our analysis. We know that for many conditions, patients may develop additional features over time, and the results of imaging or other diagnostic studies are not always available at the time when we do the exome analysis. So for this case, to demonstrate how the Moon algorithm works when there is limited phenotypic information available, we reran the analysis using only clinical features that might have been present when the patient was only 1 year old, so that included developmental delay, hypotonia, and failure to thrive, and we removed any findings from imaging studies. So in this hypothetical analysis, Moon was still able to identify the diagnostic variant in this case. And this is critical, particularly for young children and for individuals without much prior testing, such as imaging studies. For those patients that may not have a positive finding identified on the initial analysis, our reanalysis program also allows for new features that develop over time to be incorporated into the analysis at a later date. So in conclusion, Invitae is committed to providing an exome sequencing test that is accessible to all patients with rare genetic disorders and their providers. Our enhanced exome analysis now includes the intelligent engine Moon, which integrates and weighs data, and Apollo, which ensures up-to-date gene phenotype curation from the scientific literature. Our approach combines artificial intelligence-powered tools with manual clinical expert review to ensure that our analysis and reporting is personalized, objective, and accurate. And Invitae is the first and only clinical laboratory to offer the benefits of comprehensive exome sequencing with ongoing case-level reanalysis while maintaining exceptional accuracy. Thank you again for allowing me to present these case examples today. I hope they help illustrate how the integration of Moon and Apollo into Invitae's platform allows us to offer exome sequencing that's driven by experts, enabled by artificial intelligence, and personalized to your patient. Thank you.
Wow. Thank you so much, Dr. McLaughlin and Ms. Knight-Johnson. It's really great to see how so much information, both genotypic and phenotypic data, can come together in a systematic way to provide an accurate and reproducible exome. I think for me, the